Dr. Julie Niedich. She's an experienced physician who has both a personal and a medical interest in autism spectrum disorders. She's a board-certified pediatrician and medical geneticist who's worked as an assistant professor at Cornell University and appointed faculty member at Stanford University School of Medicine. Dr. Niedich has more than 20 years of clinical experience and dealt with numerous families with autism. She understands the concerns and struggles families can face and will take this opportunity to discuss the basics of autism spectrum disorder genetics as well as new testing strategies. I wanted to briefly mention that you can feel free to ask questions throughout this webinar by going up and hovering your mouse over the green sort of rectangle on the top of your screen where a chat button is located. The questions will go directly to us, and technical questions will be addressed throughout the presentation, and questions regarding autism, genetics, or anything else will be addressed in our question and answer period at the end. And with this, I will pass along the microphone to Dr. Niedich.
Okay. Hi, everybody, and again, thank you for joining us. I'm really glad to be speaking to you today about autism spectrum disorders and the genetic link. And what Diana mentioned was that I have a personal connection, and that is I have a nephew with autism. And it's been really hard for his family. So we'll talk about what kinds of tests, what kinds of genetics we think about when we, as a physician, see a family with autism, and we'll go from there. We have some learning objectives for today. The first is that we're going to be talking about just the ABCs of autism spectrum disorder, the ASD triad, what different inheritance patterns are, and recurrence risks in general. We're also going to be talking about a diagnostic strategy that includes Fragile X analysis, microarrays, and then Clinical Diagnostic Exome. Then we'll have some closing comments, and then we'll go on to your questions. So please do feel free to send in your questions about the presentation to Diana, and we'll talk about them at the end. If you are having problems with your ability to hear or see the webinar, please send those to Diana now. So onward, the ABCs of ASD. The first thing I'd like to say is that autism spectrum disorder is a category of communication developmental disorders. It isn't just one thing. However, when we group autism spectrum disorders together, there are approximately 1 in 88 children being diagnosed with autism spectrum disorders today. It is about 4 times more common in males than females, and that means that a lot of families are coming into their pediatricians or to a geneticist, genetics clinic, developmental pediatrician, or neurologist with problems that get pooled into the autism spectrum disorder group. Sometimes there are other commonly seen clinical findings. There can be sleep disturbances, gastrointestinal problems, learning difficulties, and so it's not just one thing. What we do know is that there are problems in each of these 3 spheres that affects children with autism spectrum disorders. There are problems with social interaction, there are problems with communication, and there are problems with development. They don't have to be 3 completely equal spheres for any given child, and there may be children who have better communication or better development or better social interaction for that matter, and still fall into the ASD spectrum group. There also are kids who only have developmental issues who don't really fall into the autism spectrum group because their disability doesn't affect their communication or their social interaction. Because this is a spectrum, like every other developmental issue, there are higher and lower functioning individuals who share the diagnosis. Sometimes families will start to talk and you'll find that somebody's child is particularly high functioning and somebody else's child is particularly low functioning, and sometimes it's hard to pool all of those patients together. Some affected individuals have very high functioning and are very intelligent, while others are in fact intellectually disabled. Some affected individuals have a specific ability that rises above their other disabilities. And some affected individuals have other symptoms besides their developmental or social issues. We mentioned the GI symptoms, the sleeping disorders. There also are children with who fall within the autism spectrum who have other malformations or heart defects or other internal organs that don't function properly, and sometimes those are kids who are more likely to have a genetic cause of their developmental issues. So let's talk a little bit about inheritance. And this is a really important part of looking at the genetics of autism. A lot of times when we talk to families, there's only one affected individual in the family. Sometimes when we talk to families, we find two or more affected siblings, brothers and sisters in one family, or you find generations where there are affected patients. That's why we sort of have to always, when we talk about this, look at each individual family. All geneticists will take a complete pedigree, that means the complete family tree of the patient and both parents, to try and figure out if there is a pattern of inheritance within this family. So first of all, we're looking here at two sets of chromosomes. On the panel with the boy who's labeled XY, he, in fact, has 22 pairs of chromosomes that are exactly the same. And then at the bottom, you'll see he has one X chromosome and one Y chromosome. And if you look at the panel that's with a little girl labeled XX, she has the same 22 pairs of regular old chromosomes plus 2 X chromosomes. The X and the Y, or the X and the X, are the sex chromosomes. And aside from those two, the other pairs of chromosomes are the same. Chromosomes are the packets of DNA that carry all the genes and go from parent to child. Each one of those pairs of chromosomes is inherited from a parent. For the 22 pairs of chromosomes, just looking at them under the microscope, it's hard to tell which comes from the mother and which comes from the father. However, we do know in a male that the Y chromosome has to come from the father and the X chromosome has to come from the mother. In the case of a female, we can't tell the difference between the X chromosomes just looking at them like this. In many patients who are perhaps over the age of 2 or 3, a chromosome study would have been maybe the first test offered by a pediatrician or a geneticist or a neurologist to try and see if there was a chromosome rearrangement. Oops, are we disconnected? Oh, I think we're okay. I'm not sure why that popped up, because this looks like we're still live. I'm sorry, we suddenly had something pop up. Maybe just close it, Diana. No, I think we're still live. See, it's still going. Okay. Oh, I'm sorry, I should have closed it. I didn't realize. I'm sorry. So in any case, this would have been the first test offered to a family in trying to figure out where the disability of the child arose. now we tend to use another test, which I'll talk about later, but it's based on this same principle of looking at the chromosomes. This is sort of, for geneticists, a larger view of all the DNA put together that's inherited from both parents. We'll talk more about the other studies as we go through the webinar. But basically, we have several different kinds of inheritance patterns. The first type that we're going to talk about is called autosomal dominant inheritance. In this particular diagram, you see that there's an affected father who has a disorder. It could be almost anything, and an unaffected mother. And there's a 50-50 chance in any pregnancy that the gene for the dominant disorder is going to be passed to the offspring. It doesn't matter if it's a boy or a girl with a regular autosomal dominant condition, so that you see if this family has a pregnancy, there's just a chance in every single one for this gene to be passed on, or the father could give his normal chromosome and normal gene to the offspring. If we look now at autosomal recessive inheritance, this is a little bit different because in this case, the parents don't necessarily know that they carry a gene that can cause the disorder. They're usually perfectly fine and go on and have a 1 in 4 chance in any pregnancy of passing on both of their copies of the abnormal gene. If you look at this carefully, you'll see that it really also doesn't matter if it's a male child or a female child, but what you see is that there's the one boy who's got both sides of his body colored in, and he's affected. Again, it wouldn't matter if he was a male or a female in an autosomal recessive condition. There's a 50-50 chance of a child inheriting either the father's abnormal chromosome or the mother's abnormal chromosome, and therefore you see 2 children colored in half and half. And then you have one child who's completely normal and has gotten 2 copies of the absolutely normal gene from each of the parents. So you see one affected child, one completely unaffected child who doesn't even carry the mutation, and 2 children who carry the mutation. These are chances that it'll happen in any given pregnancy, and therefore if you have 4 children, it's separate for each pregnancy, and it's possible to have 2 or 3 even or more affected children amongst the 4. In addition, you could also have all your children carry, or in fact all your children not carry the disorder at all. Another type of inheritance comes with the X chromosome. As we looked at in the slide that showed the 2 sets of chromosomes, males give a Y chromosome to their sons and an X chromosome to their daughters, whereas the mother gives only an X chromosome to a daughter or a son. If the mother carries the mutation silently that's on the X chromosome, she would have a 50-50 chance of passing that gene on to her offspring. If it were a daughter who received that abnormal gene, the daughter would likely be just like the mom and unaffected. Sometimes there's some variation in that, but for the most part, X-linked recessive disorders affect males. If a son gets that X chromosome from the mother, there is no other X chromosome to sort of balance the effect of an abnormal gene. And the son, in fact, will have full effect of having an abnormal gene. And that's the little boy you see who's completely colored in blue. The other children will have gotten an X or a Y chromosome from their father and the normal X chromosome from their mother. Again, sometimes it happens that more than one affected child can occur because it's separate for each pregnancy. The risk for having a child is pregnancy-based, and while you might say, well, the chances are lower if you've had one, it really doesn't seem to be the case when we look at families. When we look at autism spectrum disorder in general, we feel like there is some genetic influence as well as some environmental influence in almost every patient. And therefore, we're sort of combining both environment and genes into something we call multifactorial inheritance. Multifactorial inheritance is seen in lots of different disorders like diabetes, but in autism, this seems to be the case for many families, that there is a combination of environmental effects as well as genes that go into the severity of the presentation in any of the patients, as well as in whether or not there is a disorder at all in any given child. There are lots of genes that have been identified as causing autism spectrum disorders. We've listed a few of them there. Some of these are the genes that cause something called Rett syndrome. MECP2 is the most common gene that's been found in girls with Rett syndrome, but the other genes at the bottom, CDKL5, MEF2C, and FOXG1 are also related to variant Rett syndrome presentations. P10 is a different gene that causes macrocephaly or large-sized heads, as well as autism spectrum disorders. And FMR1 at the top is the Fragile X gene, which we'll talk about a little bit more in depth in a few minutes. But there are lots of genes that have been implicated in a child having an autism spectrum presentation. And so it's hard for somebody as a geneticist to pick any one gene outside of this particular group that you would want to test in any given family. Sometimes I'm asked, well, what about epigenetic effects? What is epigenetics? Well, in epigenetics, it's not a change in the DNA itself. It doesn't involve the genetic code, but it's changes in the area around the gene. This may turn on or turn off a gene, making it work better or work worse, or be found more active or less active. Through these changes, we have lots of different environmental factors that can cause them. We feel that there are nutritional effects, medication exposures, radiation exposure that affects the genes. But most of the time when we're talking about an epigenetic effect, the epigenetic effect happened generations before. It doesn't have to be in the child who presents with the disorder. It's most often, in fact, in the grandparent generation or the parent generation who've had some kind of exposure that has changed the proteins and other factors surrounding the gene itself before it's passed on to the child. Looking at this picture, you can sort of tease this apart a little bit more because this is a really difficult topic. If you look at a cell has a nucleus inside. Inside that nucleus are the 23 pairs of chromosomes, and in those chromosomes there are about 22 to 25,000 genes packaged in every single cell. If you look, though, at a chromosome, the chromosome is made up of coiled DNA. And there are a lot of proteins and other factors that make those coils happen. So if you affect the coiling of the DNA, which can affect how then the gene works, you affect the child later on. You can have effects from temperature, radiation, food, drugs, specific nutrients, and they can change the environment of the DNA so that it's not directly a mutation in a gene, but a change that can show up in the child down the road. It's a hard topic. There aren't any tests yet for epigenetic effects, so that if anyone out there is concerned that there was an epigenetic effect that caused their child to have an autism spectrum disorder, you have to wait on that. We're not ready for testing. Moving on to discuss a little bit more about recurrence risks. What is a recurrence risk? It's the risk of having another affected child or children following a diagnosis of something in the family, in this case, ASD. What is the risk if you have one child who is affected? What is the risk if you have two children who are affected? What is the risk if you have a brother or sister who is affected? Well, here are the basic facts. ASD recurrence risk has been estimated to be between 3 and 14%. However, some authors have quoted a risk as high as 18.7% when you have only one affected person in a group of children, in your own children, I should say. There is an additional twofold risk increase whenever you have more than one affected child. So once you've had a second affected child, your risk goes up to 32.2% approximately. And the risk also increases when the paternal age goes up over 50 years, although that's not something that we normally can test for. We just know that there is a risk of new changes within genes in males as the paternal age advances. So taking all of that together, what does this mean for diagnosing a genetic portion of why a child has an ASD? Well, we have a proposed strategy for looking at the genetic causes of ASD. And one of the things that we found is that many families go on what's called a diagnostic odyssey. Their doctor may do 1 or 2 tests to start with, and then go on a whole series of testing that can include biochemical testing, gene testing, other factor testing, and that can go on for months to years before anybody comes up with a specific diagnosis. Sometimes no diagnosis is reached doing all of that testing, and even with our strategy, we can find no specific genetic diagnosis. However, what we've done is we're proposing something that we think will both cut down the time of testing as well as the cost of testing in the long run. What we're suggesting is that patients start by having Fragile X testing and a microarray, and we'll discuss each of those separately. There are 2 kinds of microarrays that are currently available. One is called a 180K oligo array. The other is called an SNP or SNP plus CGH array, and we'll talk about that. That would be our first test, and that's actually the kind of testing that I've usually offered to my patients straight off the bat. The side boxes include different tests that might come up based on an individual family history. So the X-linked intellectual deficits next generation panel tests for a huge number of genes that are only found on the X chromosome. In a family where there are multiple males who are affected, or males from several generations all related through the mother, this may be the next best test to have done after the Fragile X test and some kind of microarray. On the other hand, if your doctor sees specific symptoms that make him or her think that maybe it's a specific gene, for example, your child presents with all of the symptoms of Rett syndrome, then going first for some kind of targeted analysis of one single gene is definitely the way to go. However, for many families, there will not be an inheritance pattern to look at, nor will there be a specific set of symptoms like Rett syndrome or macrocephaly with some of the other features that go with PTEN gene mutations. And maybe the best first test after Fragile X and a microarray is our first-tier exome test. In that test, we look at all of the genes that have been associated with any disease that has been reported in the literature. It is a rather comprehensive test, which would then reflect to testing all of the genes that have been found in humans, whether they've been associated with disorders or not. We do know we have limited understanding of genetics at this point, and even though our understanding has grown tremendously in the past 10 or 20 years, we don't know everything about every gene. These tests, however, don't even look at the areas of DNA in between genes, and that's something that's still really a research test only. So, as we said, step one, Fragile X testing and a microarray. These are 4 separate individuals who have Fragile X syndrome. It is syndrome that is associated with developmental issues and autism spectrum or autism-like presentations. The bottom 2 individuals are males who are already adults. The top 2 individuals are obviously young boys. Both of the young boys look pretty much like cute, normal young boys. There isn't a lot in the youngster that differentiates the Fragile X patient from other kids. There are some behavioral issues. There may be some people talk about large ears or maybe some loose jointedness. But as young children, Fragile X males usually look fairly cute. As they grow through puberty, there are other changes that happen, including enlarging testes that go well beyond what's usually seen in the maturing male, as well as elongation of their face, so that you see that sort of long look that both of the males at the bottom have. Most males with Fragile X do have intellectual deficits and behavioral issues, and most of them do have some social problems and are diagnosed with an autism spectrum presentation. This is a test that is run on the patient, him or herself. We do sometimes have some mildly affected females who can present with some autistic-like features. And then we also have testing for family members who may be carriers of what's called a pre-mutation in the Fragile X gene. This is an unusual gene where a mother may not have a full mutation and may have a pre-mutation that can be looked at. But once this is diagnosed in a family, it can be used to predict whether or not a child will be affected and can be used, in fact, right after birth or during a pregnancy. Array CGH, or a microarray, is another way of looking at all the chromosomes at once. And usually it's done instead of a chromosome analysis now because we can see pieces of DNA that are much smaller than we could see using the routine kind of chromosome analysis. In the first step, the patient's own DNA and the exact same amount of a normal control DNA from the same-sex person are mixed together, each of them with some fluorescent dye. In this case, you see that the patient's DNA is mixed with a green dye and the control DNA is mixed with a red dye. But what happens when you combine green and red and they're exactly in the same amount in these fluorescent dyes, it looks yellow. So if all of the DNA comes up looking yellow after the DNA is mixed and the testing is done, then the patient has the exact same amount of DNA at every spot as the control DNA does. If, though, the patient has a little duplication or extra DNA, it will shine green at that spot. If the patient has a little deletion or a little missing or lost piece of DNA, it will shine red at that spot. And we look at all of those spots through a computer and look for a piece that's missing or extra. It could be a whole chromosome. It could be a tiny little piece that you cannot see with the microscope. And if you look at the lower right-hand corner, you see that in this case, we're looking at a chromosome 7, and in the long arm of the chromosome 7, you see a little piece of DNA that's cut out, and that would be the red area that you see going down from the line across the plot. And the part that has the extra control DNA is on the upper part. So this is why we have sort of looks like a reflected picture of the missing area. This is one kind of DNA plot. There are lots of other kinds of DNA plots, and we all look at whatever our computer software shows us. But most microarrays are the same. If it's called a 180 oligoarray, then it's pretty much the same as other 180 oligoarrays with rare exception. If it's called a SNP or SNP plus oligoarray, they're pretty much all the same as well. These two boys were each diagnosed using a microarray. In the boy at the top, he had a little piece extra of chromosome 16 at the band P11.2. This is called the 16p11.2 microduplication syndrome. And this duplication can also be seen as a deletion. And we see it in approximately 1% of patients with ASD. There is no syndrome involved. Sometimes it's new in the patient himself or herself, so that it's not something that's necessarily inherited. And usually the developmental issues are early onset. In other words, this isn't something that's a slowly developing loss of abilities. In the 15q13.3 microdeletion syndrome, which the boy at the bottom has, there could be mild or moderate delay in development. Some of those kids have seizure disorder, and sometimes we see subtle facial abnormalities. In this boy, some people, specifically geneticists, might say his ears were a little bit cupped. There are different sizes of this microdeletion. If a bigger piece is missing, then in fact the disorder can be more profound. And if a smaller piece is missing, it's less profound. But this has an approximate incidence of about 1 in 40,000. So it's not that rare for geneticists. Now, let's talk about step 2, which is the Clinical Diagnostic Exome test. What's an exome? Well, if you look at this slide, you see that we have a picture of a cell, and like before, inside the cell is a nucleus, and inside the nucleus are chromosomes. So each cell contains the 23 pairs of chromosomes that we've talked about already. Chromosomes contain hundreds to thousands of genes each. Each of those genes is made up of exons. The exon is the part of the gene that's made into a protein at the end of the expression of that gene in a cell. So that you take all the exons for a gene and they're biochemically linked together, and then they form a protein. The proteins then tell the body how to develop and grow. And if we say, what is an exome? It's just a test that contains all the exons, all the coding regions of the genes in the whole nuclear genome of a person. What is exome sequencing? Well, here you see, instead of a double copy of each chromosome, you see a single copy of each chromosome, and some of the genes on each of those chromosomes is written out here just to show you the kinds of variability that you would see, lots of different genes all over the genome. So what is exome sequencing? It's sequencing all the protein coding region of the genome. It analyzes 22,000 genes. It's able to detect new changes that are either new in the patient or new genes that have never been described associated with disease before. And it eliminates the diagnostic odyssey of going from test to test to test without an answer. It is possible to have no answer from exome sequencing. This isn't the only test that will become available over the next, say, 10 to 20 years. But at this point in time, 2012, this is one of the ways to sort of cut through testing for months on end. So at the top in panel A, you see a timeline from the start of testing at diagnosis of an autism spectrum disorder all the way through for about 2 years. And A is what actually happened to one specific patient who we tested. He had more than just autism spectrum presentation. He also had a heart rhythm disorder called long QT. So his doctor first did the chromosome analysis and then went to the long QT genes, which I can understand completely. After that, he had biochemical testing, including urine organic acids, plasma amino acids, and some neurotransmitter tests. He then had his microarray done. He then had another biochemical test called plasma acylcarnitines. Then he got tested for something called Smith-Lemli-Opitz syndrome, which is a specific gene test. Then he got tested for congenital disorders of glycosylation. He had sequencing of a series of genes that caused those. We're already getting up to a year after diagnosis, and there still is no molecular genetic diagnosis for this child. And the parents have now had testing that has cost thousands of dollars either to themselves or their insurance. At about 15 months, he had deletion duplication testing done for congenital disorders of glycosylation. Then he went on to have the Rett syndrome genes tested, as well as another X chromosome gene tested. Then he had mitochondrial genes tested, and we haven't talked at all about mitochondrial genes, but those would be more rare causes of autism-only presentation. And then they went on to have the Clinical Diagnostic Exome testing done, and we identified a mutation that caused all of his problems. So if you follow our methodology, our methodology would recommend Fragile X testing first, with the array-based test. The array will let people know whether or not going on for another chromosome analysis is important or not. And once these two are done, the next test to do would be the Clinical Diagnostic Exome. It can take weeks to get results from that, but in effect, it will cut down the amount of time you wait for a diagnosis by many months. What is the impact on a family of having a diagnosis? First of all, it helps establish proper medical management of the child right here and now. If we know that there are medical problems that are associated with a specific gene, then we know what to help treat with right now. It also can help us anticipate future clinical findings. It will, in fact, also provide more accurate recurrence risks for all of the close relatives and future children for that couple, as well as for the siblings of the affected child. It allows for easy genetic testing for the specific familial mutation or mutations. And then there's the huge psychosocial benefit for just knowing why your child is the way he or she is. So in conclusion, the ASD diagnostic strategy evolved to complement the ongoing research and technological advances that we've seen. We've changed from chromosomes to microarrays. We're sticking with Fragile X, but it's a molecular test. And we're going to advise skipping some of the individual gene tests for a more cost-effective means of looking at a series of genes all at the same time. The Clinical Diagnostic Exome test can provide clarity for families who seek answers to explain the ASD diagnosis. And genetic answers can help tailor medical management and, in fact, prediction of what might happen to the individual child and family. Thank you very much for your time, and I'd be happy to take questions.